Thank you, Jeremy, for the introduction, and thank you, the organizer, for inviting me and speak about some of the previous experience with the MHC2 processing and presentation. And so I also had to join some of the colleagues that didn't talk about their current work, but I will provide my perspective on the problem of antigen processing and presentation by MHC class II molecules and how that fits with what we know is integrated within the in-silico prediction platforms and how we can possibly add to those platforms for a better performance. So the great things of being the second to last speaker is that I really don't have to talk about this, right? Because if we don't know it, we heard it at least 3 or 4 times in the last couple of days. So I will primarily focus on the crux of the process here, the presentation of the peptide to CD4 T cells by MHC class II molecules. One aspect that has not been really addressed, though, is, sorry, I'll start my timer so I'm sure that I'm not going overboard, is the problem of immunodominance, which falls within the larger context of immunogenicity. And so what we know is that if you take a complex antigen and you look at the number of peptides toward which a CD4 T cell response is organized, you will find that only few peptides from that larger antigens are actually the focus of such response. But the interesting thing is that within that antigen, there are peptides that you will be able to bind to the MHC2 expressed by that DC, and yet they are not recognized, or they are not interesting to the CD4 T cell compartment. So the question is why certain peptides are actually selected for presentation and why others are not. And obviously, if we can address that question, we can somehow address some of the issues that pertains the recognition of microbial antigens, the engineering of vaccine, autoimmunity, and all the other nice things, including immunogenicity. That, of course, requires immunodominance for its occurrence. And so, obviously, we look at what happens with an antigen-presenting cell, and it's really the textbook way of looking at MHC2 antigen processing and presentation with the internalization of the antigenic protein within the APC through the endosomal compartment, and the antigen is experiencing a increasing acid and low voltage environment where catepsins can function as proteolytic enzymes, and again, as a textbook, view of the process, we think that catepsins are cleaving all the peptides of those antigens, and then within the M2C, the MHC class II compartment, MHC class II molecules that have been synthesized as an alpha and beta chain in the endoplasmatic reticulum through the Golgi, they reach the M2C, and that's where then everything happens, right? MHC class II are synthesized, and they basically harbor the clip peptide, which is a kind of a placeholder and they help with the refolding of the MHC class II. And in the presence of HLA-DM, most of the MHC class II alleles release the clip peptide, and then they start this peptide selection within the endosome. And then through some process that we're trying to understand, right? Few of these peptides are selected for relocation to the plasma membrane and recognition by CD4 T cells. So the question is, can we predict the outcome of this process? And so the problem, though, is that, as many of you have illustrated in today and yesterday, we have shrunk the problem of epitope prediction to the bare bone of binding, thinking that at the end of the day, something binds, pretty much it will be an epitope and will be recognized by CD4 T cell. And then, relying on the binding prediction, then we say, okay, we have identified CD4 T cell epitope. Well, evidently, it's not that easy. I think that the human being like to take complexity, reduce it into simplistic, explanation, and then as long as that is good enough, then we take that and we forget the complexity from which that was derived. But really, when you're trying to look at what the in-silico predictions that are trying to provide the answer of binding are doing, is pretty much a prediction ball. and I really don't trust wizards or witches, and neither should you. So this is 2011 paper. Things have changed a little bit since then, but not that much. If you look at the publication from the Deutsche Nova group, they have been fundamentally focused on DP and DQ, but they also have done some work on HLA-DR. And so they consider the epitope prediction with the, you know that there is a 1%, a 5%, and a 10% score. And obviously, depending on how high you go with that threshold, you're going to incorporate more and more binders, but possibly also more and more non-binders in that prediction. So they stop at the top 5 percentile based on that scoring in one of those 5 prediction platforms. And what they found is that of the experimentally identified epitopes, NetMHC2-PEN and NetMHC2 would basically pick 55% of those. The other perform more poorly. If you take those that were identified as binders by NetMHC2 or NetMHC, only 10% of those end up being true epitopes. Okay, so we are looking at how sensitive these platforms are and what's their positive predictive value. Okay, you can tell me, well, from 2011, things have changed. Remember that, these predictions are as good as their training data set. And obviously, from 2011, the data set has increased from about, this is the cumulative score, so from cumulative size of the database, from 45,000, we got to about 75,000 or so. And here they show over time how many references they have included in their training data set and the number of data sets. But then when they have recently re-established the efficiency and the sensitivity of their platform, they identified NMHC2-PEN as the best performing one, both if you look at the Spearman ranking correlation coefficient as well as the AUC of the ROC curve. So NetMHC2-PEN still is fairly better than anything else. And yet, if you compare NetMHC2-PEN and the consensus from the immunoepitope database, and you look at how well they perform against the experimental epitopes that are identified in ELISpot assays. So what here you are looking at are the cytokine spots every million cell in the ELISpot assay, and on the y-axis, both for the in-silico prediction and the consensus, they're ranking. And they've done in such a way that the greatest is the ranking, the strongest is the peptide. Okay, so usually when you look at the scoring, is actually the opposite, but they wanted to do it in a way that was proportional. So on this side, you will have the predicted but not found in the ELISpot. On this side, you will have the false negative, those that are not predicted, but they actually were found. In here, you will have the predicted and found, and in here, you will have the true negative, the non-predicted and not found in the ELISpot. And you can see that there are a considerable number, even for alleles for which the data set are quite large, such as DR1, you still have a large number of predicted but not found. So you have, as we mentioned before, the false positive problem, but you also have the false negative problem. And when Chavez, and he's from the Andrea Ferrante group, they have done they have come out with 2 or 3 publications trying to, again, establish what is the efficiency of the in-silico prediction, particularly the IDB and NetMHC2. Their conclusion is that, yes, it can facilitate epitope discovery, but all share a high degree of false positive and also false negative prediction. And there is, if you look at the IC50 or the KDs, they are calculated by the predictors. There is still disparities among them and with respect to the actual value. So where do we go from here? The problem is there are several aspects that make it difficult to predict immunodominance and therefore immunogenicity. So I will quickly go over a few of these. I will say those that I've been investigating more in depth, and then hopefully they will trigger some discussion both here and out of here as to how we can incorporate some of these aspects in the next in-silico prediction algorithms. So one problem obviously is the fact that MHCs are highly polymorphic, and we know how polymorphic that is. It's the most polymorphic site locus in a human, and the polymorphism is located in the binding site, which makes it an issue in terms of prediction. And also the problem that there are alleles for which there is a very little binding data available, and this is what these predictions are based on. The other aspect is the fact that the frequency of alleles across the human population changes depending on where you are. So, if we're trying to figure out what's going on, for instance, in the U.S., for the U.S. population, well, that not necessarily will apply to Japan or to the ex-U.S. population. And so this is a quite nice visual way to see that, if, for instance, if you take DR beta 1 in Australia, there will be the 2% coverage. If you take that in Norway, it will be the 9%. If you consider that in, within the Mexican population, it's 7%. So it changes, right? And everything goes accordingly. When you take your in-silico prediction, usually they're not weighted unless you do a weight by yourself, right? And the other aspect is that, as I was mentioning, even for the training data sets, for instance, of the NetMHC2, their sizes are different. And obviously, if you're looking again to the most frequent alleles in the US, you are pretty much covered, okay? So you got your 0101, 0401, 0701, 0901, etc. So, having a larger data set, you think, okay, that's a little bit more reliable prediction. But then there are aspects, for instance, of alleles that are in Japan, such as the 0803, which is more frequent than 0801 and 0802. So now, if we're trying to figure out what's going on with our sequence, and trying to predict potential immunogenicity in a Japanese population, then we're missing this one. Or we're missing, for the same reason, 1503, which is more frequent than 1502 or 1501. 1503 has the same frequency as 1201 in the US population, so they are both about 2 to 3%, and yet we have basically no data for 1503, and we do have plenty of data for 1201. So also within the US population, our prediction is biased because of the different size of those data sets. And what about 1202? 6% of the population outside US and Japan. We just forget about, 450 million people just because of that? Okay. The other problem, which is less in terms of frequency and more intrinsic to the peptide binding per se, is the fact that peptide binds to MHC molecules in a flexible fashion, and that's usually not considered in this in-silico prediction, except a couple, the NetMHC2 and the NN and SMM, they do. Otherwise, you will see that that doesn't happen. So, of course, you know the structure of an MHC class 2 molecule. This is the HLA-DR3 with the clip peptide bound to that. And I think all you know that we postulate the binding of the peptide to the MHC binding site rely on hydrophobic pocket interactions, the so-called anchors, and then an extensive hydrogen bonding network between non-polymorphic, between the side chain of non-polymorphic amino acid in the helices and the backbone of the peptide. And so we usually think that this is a non-polymorphic or non-allele dependent source of binding energy, and this is an allele-dependent source of binding energy. So because of that, most of the predictions have relied initially on identifying the pockets, hydrophobic pocket interaction. So if I have a sequence and I run the sequence through my predictor and I find that there are at specific location amino acid that will fit nicely within those pockets, then okay, I'm off the hook. That peptide is binding. If not, not, right? And so the prop-red and the epitope, based on the epitope training data set, and then the MHC, these are all basically pocket profiling. And again, the idea is I'll make sure that I have 4 amino acids that fits those 4 pockets for every given allele, and I'm okay. Well, this was a killer experiment in 2003. They synthesized a set of peptides from DHA hemagglutinin 307-319 of H3 influenza, held the anchoring amino acid constant, and then changed other 4 amino acids that were in making hydrogen bond contact, and off you are. If you consider the wild type, the one with the with the red black dot, and then this one with a triangle, which has again the same anchors but 4 different amino acid at position 2, 3, 7, and 10, you basically lose between 4 and 5 log of affinity. Same anchor. So evidently you cannot rely on your anchors, right? There is something more to that. So someone who was smarter decided to look at something, to add something more to the prediction, constructing those that are known as a position-specific scoring matrices. And pretty much they look at the full length of the peptide, right? So they assign a value of binding. I would say binding contribution to every position for every amino acid at that position, right? And so by doing so, they consider if I have, for instance, an alanine epi1 and a valine epi2 and another amino acid epi3, so for the full length, what I'm gonna get, which means that we pass over the problem of only anchors and we look at the full length of the peptide. But one aspect here is that every amino acid is looking at its own individual contribution to the binding. And this is a problem because the binding of a peptide to the MHC is not due to single individual contribution. You got to think like this is an allosteric system, like hemoglobin with oxygen, okay? Instead of having oxygen, you get the peptide amino acid, and instead of having hemoglobin, you have the MHC binding site, but it's the same system. So if one amino acid binds, it helps other amino acid to bind, and if one amino acid does not bind, it will prevent other amino acid from binding within that sequence. And so this is a cooperativity, which you can call, flexibility or allosterism, if you wish, versus affinity curve. And what this is saying is basically that there is a range of affinity within the peptide MHC2 system in which the MHC2 is flexible enough that it can sort of overcome the lack of binding energy from the peptide to the point that it can still form a complex. But then there is some sort of a energy threshold, and past that, any elimination of an individual source of energy will have an exponential effect on the loss of affinity of that peptide. So it's not simple that, I have amino acid X here and amino acid Y there, and I can look at their contribution independently. Things are actually more complicated than that. And this is also visible in some molecular dynamics simulation that was run by the Larry Stern group in Worcester, UMass. And again, this is a molecular dynamics simulation because it's very tricky to actually crystallize the empty form of the MHC2 molecule, but you can still do some simulation on that. What they've shown is that there is a significant change in the structure of the binding site when you look at the peptide bound versus the empty MHC2, which is indicating the fact that this is a fairly flexible system, right? And most of the flexibility is located in this portion of the alpha helix of the alpha chain of the MHC2. When we looked at the binding of a couple of different peptides to a couple of different MHC2, we actually found the same we came to the same result basically through a different way, by looking at the thermodynamics of the binding instead of simply looking at the affinity and off rate, which is the standard way to look at peptide interaction with MHC. What we found is that you can have different peptides binding to slightly different MHC2 molecules. And their affinity can change within 1 log, which is significant, but not as great within also scheme of things, and yet their thermodynamic mechanism will be different. That's why, well, going back to what I was saying, because of being a flexible system, the MHC can overcome the loss of enthalpy with an increased entropy. By entropy, we can go back to flexibility, and enthalpy, we can think of the binding energy. I put a little vignette here if you forgot your thermodynamics from college. And the same thing when we ran a similar molecular dynamics simulation as Larry's did for these other peptides and these other MHCs, we found exactly similar results, right? That you have increased flexibility, shown here by the blue lines, when you have a peptide that is lacking some of those sorts of interactions, whereas those peptides, such as the wild-type peptide, is basically locking the system in a more structurally rigid conformation. Why I'm saying all of these, you say, well, why are you telling us about, conformation of this complex and all these sort of things? We want to know about immunogenicity. We want to know whether that peptide will be present or not. Well, one aspect is that depending on the peptide that's bound, the structure of the molecule, the flexibility, and the structure of the MHC peptide complex changes. And so in the complexes that harbor the peptide where we took away some of the interaction hydrogen bonds, you have an increased flexibility at the alpha chain portion, exactly like Larry Stern would show in his empty bound molecular dynamics simulation. And the structure and the flexibility will be instead more rigid and more compact in the complex harboring the wild-type HA peptide. And now I'm telling you why that's important. Because when you look at this other guy, HLA-DM, which is, as we know, the arbiter of the epitope selection process in a dendritic cell or any APC, right? So that's the one that kicks the clip off the binding site and facilitates the exchange of antigenic peptide from the binding site. Well, number one, DM physically engages the peptide MHC2 complex, okay? And the other things that we know, it engages the peptide MHC exactly in that location which is most affected by that flexibility. So if you have a peptide which determines how flexible and the structural rigidity of the complex, it will be more or less affected by DM activity depending on how it binds to that. Okay, so that's why I need to incorporate that thermodynamic structural aspect to the story. And so when we looked at the ability of DM to release the peptides, in black and in green, you have the HA peptide bound to two different MHCs. And in black and in green, you have HA peptide in the absence or in the presence of DM bound to the wild-type DR1. And HA, we know, is fairly insensitive to DM activity. And then we have the other peptide, which has fewer interaction, has already a shorter half-life, but in the presence of DM, the half-life of that complex is greatly reduced. And the thing becomes even more magnified when you look at a MHC2 that where we made a mutation in the structure to actually increase that susceptibility. And so when you plot how flexible is the complex versus the activity of DM, you see that the more flexible is the complex, the more DM will be active on it, right? So DM is sort of discriminating complexes within your dendritic cell based on their flexibility. And this is another way to look at the same phenomenon. first of all, DM works at acid pH, in acid condition. So as we go from the pH 6.5 to the pH 5.5 to sort of mimic the progression of the endosome within the APC at reducing the pH level within the endosome, the activity of DM increases, and increases in which way? Well, basically by sort of a flattening down that allosterism affinity plotted that I showed you before, which means basically that DM is facilitating survival of complexes that are rigid, right? Like the one I showed you where you have HA bound and not the mutant. So if you take all these aspects together, what I'm trying to argue here and what we have shown actually is that as you move from a peptide, which is rigidly bound to a complex, right? You basically can still generate complexes, even with peptides that you will not expect to be able to bind, because again, the system is flexible. They will compensate any missing interaction from the peptide up to a certain point. So this, after that point, then you got an exponential lack of folding or loss of binding. It's the same thing. One aspect is that the DM will be more interested in getting rid of such flexible complexes as compared to those that were formed in a more rigid fashion. Now, one aspect that is missing into immunodominance and immunogenicity is, okay, how these things are processed. And we usually forget about it. So the aspect of epitope generation by catepsin that also, up to this point, has not really been addressed in silico prediction. So we're missing DM, which is not included. We're missing in silico catepsin cleavage, and we're missing all the different aspects of HLA frequency distribution. So as we mentioned, the textbook way to look at this is that catepsin is cleaving off the peptides from the larger antigen, and then those peptides will attempt to bind to MHC and form a complex. So even keeping, even if we hold this view, simplistic view as true, we still need to address how they work and also their distribution. So Sophie made a great point about looking at B cells versus dendritic cells versus macrophages, another aspect, because the distribution of the catepsin are, we have as many catepsin as the letter of the alphabet. And their distribution is different depending on the cell you're looking at. So B cells express catepsin that are different from dendritic cells. And it's very interesting because they have a fairly even expression of some of these catepsin, S, L, B, V, and then if you look at B cells, they change. Macrophages change too. There have been a couple of very beautiful reviews by Elizabeth Mellins from Stanford, how different macrophage have different set of catepsin and processing pathways. So their actual repertoire changes because of that. And depending on the catepsin you're considering, their cleavage pattern will be slightly different. So for instance, catepsin D and catepsin E, they are aspartic endopeptidase. They like to have an hydrophobic or an aromatic, before the excision site. The cysteine and peptidase, such as catepsin C and H, they don't like to have proline either before or after the cleavage site. They don't like to have lysine or arginine at the second amino acid before the cleavage site, but they would rather have hydrophobic at -2 and -3. So you see different catepsin have different pattern, and you have to account for that, because what happens within the endosome is a serial activation, depending on the pH and the redox potential of these different catepsin. They will work first in series and then combine in a mix to generate the library of peptide that will bind to the MHC. So somebody tried to replicate that in a cell-free system, and that is the Sadek Nasseri group down in Johns Hopkins. What they've done is basically co-incubate the MHC class 2, in that case it was HLA-DR1, which we know fairly well, the M, the antigen, and they use few antigens, something from influenza, collagen for as a self-peptide for replicating autoimmunity, and then they added three different catepsins, and they looked at basically what would be the peptide library displayed by the DR in a MAPS-like assay without a cell, pretty much. So it's like doing a MAPS without a cell. And so when they actually eluted the peptide, well, first of all, they showed that this system is a fairly good replication of what you will get from a MAPS. It's not a one-to-one, but it's okay for now. We are missing many components. We are missing GILT, which opens disulfide bonds as a thioreductase. We are missing other endopeptidase. We are missing the whole aspect of activating catepsin at different time point because of pH goes down and redox goes down. But nevertheless, they've been able to generate a good amount of data showing one intriguing aspect of processing a presentation, which usually is not in the textbook, which is this, right? It was also an old idea, but it was never substantiated. The possibility that peptide bind to the MHC first, and then they're cleaved by the catepsin on the flanking sides, versus a situation where first you cleave all the peptides of the antigen, and then they bind to the MHC. Why this is important? Well, if you imagine that there is a catepsin cleavage site in the middle of an epitope, right? And you first bind, and then you are cleaved at the flanking sites, then that cleavage site is protected by the prior binding to the MHC. And so now you got a binder there, right? It's actually still there in the MHC and can potentially be presented to a T cell. Versus the situation where if you are first dependent on catepsin cleavage, then something which could actually be a binder and potentially be predicted by your IDB, Epivax, or whatever, but it does have a cleavage site internal, now you're losing it versus keeping it, okay? So the aspect of catepsin cleavage is crucial, in that it's indicating whether potential epitopes based on binding prediction are actually still viable past the cleavage, portion of the processing. And obviously, they still have to survive DM screening, right? So you bind as a, you bind before cleavage. The catepsins are cleaving the flanking sites, but then DM is still there. So you can still be screened off between the other peptides because you don't survive DM activity. In the other scenario, you first bind, you still had to survive DM screening to actually be presented, okay? So DM is always working, but it's working after two different situations. And so the question is, well, okay, so the question is how we put all these things together, right? And so one aspect that, as I said, the textbook is always the textbook, so it's a great way to start, but not always, it's not always a perfect representation of what actually we know happens in the cell. Not even of the truth, but what we think we know happens in the cell. So Larry Stern and Laura Santabroggia have done an amazing job about getting disease from lymphatic vessels and then running maps of those diseases and using a bunch of peptides. What they found is that the peptides you get off those maps from those diseases, their sequence do not match what you would expect from cleavage of catepsin, regular processing, regular pathway through early endosome, late endosome, and then presentation. And so they have somehow repackaged some of the things that we already knew, or they have proposed new pathways of processing and presentation. And obviously, immunology is complicated from the textbook already, but we wanted to have it even more complicated, right? So you can basically have processing at every stage of the pathway. And theoretically, at any point in time, as long as you form a complex, you can still be shuttled up to the membrane. Okay, so you don't have to go through all the pH level. You don't have to go through all catepsin cleavage. You don't have to go through all the DM activity. Most of the time you do. So most of the peptides you will find possibly come through a deep compartment, so they have undergone the whole process. But there are a few peptides that don't. And some peptides from influenza that Andrea found in some of her work in the mid-2000s actually are such peptides that possibly have been presented after a early recycling or early presentation without necessarily going through the whole thing. Okay, so something to account for is the activity of DM that changes depending on where things happen, and the activity of the catepsin that change depending on where things happen. Okay, but so one thing that has been already put forth, I think Joachim was talking about it, right? Using some of this method to actually feed the predictions, because if binding by itself is not sufficient, maybe we can start to learn something more by either running maps or more of this cell-free processing and presentation, which is, can be automated, is scalable, and use the data from these to actually feed the in-silico prediction. And it will start to sort of give a little bit more of information as to what realistically is processed, and one thing that I learned, I think 4 days ago, I was preparing the presentation, I actually came across the most recent paper from IDB, and I really wanted to share with you because I jumped this high when I read it, is that they actually added the MHC2 cleavage motif prediction from the database that I think, Sophie, you were showing about the eluted sequences, right? Those 60,000, 70,000, right? And they are both class 1 and class 2, but they have used 14,000 of those after a big filtering work to actually train a prediction for naturally processed ligands. And so when they try to predict new MHC2 ligands, only based on that, the predictive power was okay, it was 0.769. But when you put the cleavage, basically the cleavage motif prediction on top of the binding prediction, then the performance is way superior. Okay, so we passed the 0.8, gold mark. Something to take into account, as we move forward into this idea of a feeding prediction platform from, real life data. And the problem, though, is that they do not identify well the epitopes when we define epitopes based on CD40 cell reactivity, not based on MHC2 ligand. Okay, so be aware of the fact that if you're thinking of identify what would actually trigger a T cell, things are still tricky, and you know why, right? So, but I'm here to explain. So the whole take-home message of all this blabbing here is that antigen processing and presentation is a complex process and involves HLA2 frequency in the population, multifactorial antigenic cleavage, pH and redox-dependent effects, DM action, flexibility and structural rearrangement involved in the peptide binding event. The in-silico prediction cannot account for all the factors involved in immunodominance and immunogenicity, but only for the binding component. And so what we see starting, right, is the generation of large training data set from single class 2 allele cell-based or cell-free maps-like platform. We support development of more relevant algorithms. And so I have to acknowledge everybody who has worked on some of these analysis from University of Alaska in Fairbanks, but also especially the graduate students and the graduate students have done the bulk of the work. Also, my colleagues from Lilly in San Diego, who didn't do anything on this analysis, but they allow me to be here, and that's great. And then my previous colleagues from the Blood Center of Wisconsin, and obviously ProImmune for organizing this, supporting some of the reagents, and for hosting this talk. Thank you, and I will respond to any questions.
Well, Andrea, I mean, that blobbing, I think, as you described it, it's made my brain hurt, because not only did I realise how much I must have fallen asleep in those physics lectures, I also realised that you've just sort of thrown in an extra few layers of complexity to all of our lives. But thank you anyway.
You're welcome.
Thank you very much. No questions?
It's slightly maybe off topic, but not so much. It's just a question you say you kept saying textbook
yeah.
For the cartoons, and where if you were to try and colocalize a protein that is being processed with a compartment where it actually is happening in terms of either the processing or the loading on the MHC, which compartment would you target? Because we've been reading a lot about this and find it quite difficult.
So what you're trying to say is that whether there is a way to follow the life of an antigen.
If you wanted to follow through the processing, where it's cut, where it's loaded. So we've got this textbook, really nice textbook cartoons, but when it comes to the compartment, it seems to be not that clear.
Yeah,
blur. So
I agree, and there is so much confusion. I think that the one that did the most of the job in that is Emilio Nanua. So if you look at the work that Emil has done in trying to identify what happens where, and characterize the morphology of the different compartments, I think he has done most of the job. So I'm not able to say all the things that he's done, but I would refer to Emilio Nanua literature and references to actually dive a little bit deeper into which compartment does what, and the characterization from the morphological standpoint, the biophysical characterization. But the amount of unknowns that we have is like incredible. I was talking to Laura Santabrogio about a couple of months ago. We were at the AI meeting, and she was telling me, we have talked about class 2 like if we know everything about it, and we don't know anything about it. But the problem is that, even from the academic standpoint, the amount of support that has been done to project, of basic research looking at these things have gone down over years, favoring other areas of immunology, which is great, right? But now we are, it's a blanket, it's too short. So if you put it on one side, then, your feet gets cold. So now MHC2 is getting cold because there hasn't been so much done to understand some of this processing that you're talking about. Yeah. But yeah, Emil is the best one for that.
So you showed in the beginning studies that lack of correlation between in-silico and T-cell epitopes. And the output were ELI spots, right? So basically you're looking at a T-cell response.
Yeah.
Yes. Were there do you know anybody looked at just binding? Like just ELISA binding? Because, all immunologists, I think here, we look at these in-silico algorithms and nobody expects, to get you a T-cell epitope. We all understand that this is just tells you what is able to bind. This is, and replacing you going in the lab and running an ELISA on MHC, right? So I'm just wondering, do you know, because, the studies in the beginning were a little bit misleading because, in silico algorithms that predict immunogenicity, no, I'm sure everybody agrees. Nobody agrees to that definition. We agree binding, epitope binding, right? So, is there any studies that look at correlation between these algorithms and like binding per se?
Yeah. So the Deutsche Nova was done with binding, wasn't done with T-cell spot. The T-cell spot was the last one from Andreata and Sere and Lund, where they look at which one of the predictors that are in the IDB perform better. And now as the NetMHC2 pan works better than anything else. But the Deutsche Nova, there was the older paper, the one from 2011, that was done with binding. And the amount, yeah, but I mean the amount of there is a significant number of papers that have been done with binding data as a comparator. Now, I'll try to think. So if you look at what Larry Stern has done, if you take the 2000, so there are, I think, a paper in 2011 again, 2012, he was looking at vaccinia virus immunization, but he was actually taking apart the peptide from an antigen in vaccinia virus and was doing the binding without HLA-DM, off rate, with or without HLA-DM, and the prediction from the IDB. And you can look at the correlation with what he does when then he look at the early spot. So you have the whole, gamma of assay. And what turns out to be is that if you run the binding assay in the presence of HLA-DM, that's the best predictor for then what you see in the early spot. But if you try to backtrack the prediction, the binding prediction with what happens in its binding assay, there is a discrepancy. I can't remember how much was it, but yeah.
Just a sort of quick thought at the end, talking about MAPS assays and, the peptides that come out and how you could sort of potentially train algorithms with that. I mean, my understanding about NetMHC is that they're also still trying to, although it's a sort of neural network approach, they're still trying to predict as a position-specific matrix at the end. And there's a question whether that's, as you say, is appropriate at all at the end of the day, whether sort of a position-specific matrix is the right output when there's sort of more complexity to this. The other thing, of course, with the presentation data is it doesn't give you any affinity. It just tells you all this stuff is there, and then you make of it sort of, what you will. So it doesn't follow the classical model where you have a training set where you have a predetermined affinity from some binding assay, and you can sort of train off that to try to repredict that sort of affinity. So the question is then, if you take that antigen presentation data, which has undergone all of these sort of steps and all the complexities potentially that you've described, what sort of machine learning approach should you take to sort of educate your algorithm? Should you sort of take into account that some things about the sort of cascade of events that happens and the sort of, biophysics of that, like, the cleavage sites, etc.? Or do you want to perhaps use a machine learning approach which starts from a complete blank sheet of paper and just some abstract AI model that goes through several stages of prediction and doesn't end up with a position-specific matrix, but probably comes up with an algorithm that you can't, it doesn't have a sort of underlying, let's say, concept that you can follow how it sort of in the end will make its prediction, because that's sort of in the background of how it's trained itself.
Mm-hmm.
So I think that's a question that will ultimately come out of this, is what's the best way of, the data are there to train things with it. The question is what training approach and what learning approach should really underlie this when some of these sort of matrix-based approaches are really, I think you can take it up to the sort of 0.8 predictivity, but beyond that they will always fail. I think because they simply don't, the sort of lower dimensional projection of what really happens, and so they will never elevate beyond that sort of level of relevance. And maybe sort of if you have a large enough antigen presentation data training set and the right AI approach, you could get to a better prediction.
I'm not sure I can tell you that, otherwise I would have to send you the bill home and get paid for the consult. Okay, so yeah, I get your question. But it's a tricky one, right? And it kind of depends on what you want, in the sense that you can approach the problem in a very utilitarian way, where you say, the only thing I want is tell me what a DC will spit out after chewing on an antibody. Well, at that point, I just make a very large size training data set of peptides from, homozygous cell line that can continuously provide peptides. Do my training on that one in a very agnostic way, right? Except for the HLA and the sequence at the entry, and that's it, right? That's the only thing you want. I would like to have a little bit more control, not because I'm a macro manager, but I like to understand things, and then if I understand something, then I would like to use that understanding, right? So I would overlay different predictions. So the cathepsin cleavage is not included in the final product of option A, because option A gives me only, as they are doing now at IDB, the cleavage motif at the extremities. I have no idea if there is anything in between, right? Because I cannot predict that. But if I can have a cathepsin prediction, then I can predict also that. And so then at that point, I can start stacking up all my predictions. Now the question is, if I have option A and if I have option B, and if I've done my work correctly, then I would have option B being a nice recapitulation of option A, everything else kept equal, right? So if I'm just considering the flanking region and not in the middle, and the binding and all the rest of it can actually get the result from option A as an option B, right? And so if I had to go with my heart, I would bet on option B. If I had to go with my wallet, I would bet on option A because it's a much cheaper and a speedier way to get the results. It's okay as an answer? Okay.
Well, thank you very much again.
Thank you.